Tools and analyses for Alzheimer's disease research. The following graph portrays how many Americans over the age of 65 are currently affected by Alzheimer's disease, along with future projections. In 2015, there are around 6 million people diagnosed with Alzheimer's, with a projected increase to around 12.5 million by the year 2050. Between 2000 and 2008, death rates for other major diseases such as HIV, stroke, heart disease, prostate cancer, and breast cancer have all declined. However, Alzheimer's is the only top 10 cause of death without a way to prevent, cure, or even slow its progression. Alzheimer's disease is a chronic neurodegenerative disease that usually starts slowly and gets worse over time, with its earlier stages characterized by short-term memory loss. That's very important. And as the disease advances, symptoms can include uh, I remember now. Okay. Symptoms can include problems with language, disorientation, mood swings, loss of motivation, and behavioral issues. And gradually, bodily functions are lost, brain size is decreased, ultimately leading to death. Alzheimer's disease has been identified as a protein misfolding disease, or proteopathy, caused by plaque accumulation of abnormally folded amyloid beta protein. Plaques are abnormal clusters of chemically sticky proteins called beta amyloid that build up between nerve cells. The most damaging form of beta amyloid may be the groups of a few pieces rather than the plaques themselves. The small clumps may block cell-to-cell signaling in synapses. They may also activate immune system cells that trigger inflammation and devour disabled cells. Alzheimer's disease is also considered a tauopathy due to abnormal aggregation of the tau protein. Tangles form inside dying cells. Tangles are twisted fibers of protein called tau. In healthy areas, tau helps to keep the transport system on track. However, in areas where tangles are forming, the twisted strands of tau essentially disintegrate the transport system, so that nutrients and other essential supplies can no longer move through the cells, which eventually die. In a study conducted at Stanford University, Kay Tai and her colleagues applied the principles and methods of optogenetics to Parkinson's disease, a neurodegenerative disease similar to Alzheimer's, in an effort to decrease its detrimental effects. Deep brain stimulation, or DBS, in the subthalamic nucleus, a part of the basal ganglia circuit, and in other targets has shown remarkable therapeutic effects for treatment of the motor impairments associated with Parkinson's disease. These data suggested a model for DBS treatment in which axonal pathways are the most effective direct target of control. As seen in the graph to the left, between times 0 and 1 second, activation of D1R expressing neurons silenced local basal ganglia activity and increased ambulation, meaning that it decreased synaptic firing and transmission. As seen in the graph to the right, between time 0 and 1 second, activation of D2R expressing neurons increased this local basal ganglia activity and enhanced immobile behavior. Black bars indicate the duration of illumination. Tai was also able to manipulate synaptic transmission, presenting optogenetics as an effective way to approach neurodegenerative diseases. At the University of California, Dr. Esther Crook-Magnuson used a mouse model of temporal lobe epilepsy, online seizure detection, and responsive optogenetic intervention to investigate the potential for cerebellar control of spontaneous temporal lobe seizures. The researchers targeted a specific brain region integral to the development and cascading effects of epilepsy, such as the hippocampus and lateral and midline cerebellums, to find a correlation between epilepsy and optogenetic approaches. While interventions in which light was delivered to the hippocampus or lateral cerebellum did not consistently affect the frequency of seizures, when light was instead delivered in 3-second pulses to the midline cerebellum in the PV Chenorodopsin 2 animals, seizure duration significantly decreased. This is shown in Figure A, with the blue bars representing the light-exposed mice. The percentage of mice experiencing no seizures at all increased from 41 to 92%. There was also a significant increase of about 65% in the prolongation of the interseizure interval, as shown in Figure B, turning out to be a therapeutically desirable outcome. The field of optogenetics deals with the ability to use light to control neurons that have been genetically sensitized to light. At the Taub Research Institute at the Columbia University Medical Center, Professor Syed Abid Husseini and his colleagues are currently researching ways to apply the methods of optogenetics to Alzheimer's disease. In order to employ optogenetic approaches, 4 steps must be followed. First, light-activated proteins are required. By exogenously expressing light-activated proteins that change the membrane potential in neurons, light can be used as the on-off switch to control behavior of neurons. Naturally occurring genes, or opsins, are covalently bound to a chromophore, retinol, which, upon absorption of light, isomerizes, activating the protein. The first of these neural switches is channel rhodopsin 2, or CHR2. When expressed in a neuron and exposed to blue light at 470 nanometers, this non-selective cation channel immediately depolarizes the neuron and triggers a spike. Light stimulation of halorhodopsin, or NPHR, a chloride pump, hypolarizes neurons and inhibits spikes in response to yellow light at 583 nanometers. Genes coding for these light-sensitive proteins can be delivered to the target cells by transfection, viral transduction, or the creation of transgenic animal lines. Y-field illumination can be temporally controlled using an ultra-fast shutter with a constant light source, fast switching of an LED, or one-photon laser scanning microscopy. All cells expressing the light-sensitive proteins in the light path will be stimulated in concert. Electrodes can be used to monitor the effect of changes in membrane voltage. Many fluorescent-based biosensors with compatible excitation profiles, some of which are also genetically encoded, can be used to measure different cellular readouts. Professor Husseini is treating Alzheimer's like a cancer. Treat it early before it spreads. Alzheimer's is postulated to begin in the entorhinal cortex, as shown. Through imaging within this used electrode, Husseini can see a slice of the brain in essence, focusing on the hippocampus and the entorhinal cortex, drawing his data from these brain regions. The top image is a slice of a mouse model brain obtained through an electrode. The bottom image is a targeted region, the hippocampus, with fluorescent expressions shown to quantify synaptic transmission. This is the desired image outcome. The following is a selected bibliography. Thank you for your attention.